I think this is a really important question because a lot of people leave this class and they're like, oh, I'm going to be a doctor. I'm never going to learn organic chemistry. But turns out drug molecules are organic molecules, amino acids and peptides and proteins in the body, all organic molecules. Sustainability, right? Breaking down plastic, those are all organic molecules. Solar cells, a lot of those are organic molecules for sustainability. So I'm just curious what you all think, and what you can maybe take away as some of the applications of organic chemistry. And this is really important. Science education is super important, especially right now. A lot of science funding is getting cut, but all of us sort of can be responsible and understand how important science actually is. And that is part of our everyday, right? If you're in food sciences, everything going on in the food that we eat, mostly organic molecules, right? Carbohydrates are carbon, oxygen, and hydrogen, right? Protein, those are all built of amino acids, which are organic molecules. It's everywhere around us. All right, so we have some results. So go ahead and see what you all think. All right, yeah, medicines, drugs, pharmaceuticals, in your body, chemicals. Yeah, there's a lot of bad chemicals out there too. I think it's quite important to acknowledge that, right? Food sciences. Some people, it seems, are thinking about, solving problems. Bioengineering, absolutely. That's a huge part of organic chemistry is in that as well. We see a lot of human health, pharmacology. There's a lot of things that we see here. Doctors, cellular mechanisms, medications. These are all super, super important things that organic chemistry does. So if you take something away from this class, this is a class in problem solving, and it's also a class in a science that is, chemistry is called the central science. I think it's quite important. All right, so we'll see. It looks like people sort of stopped responding so much here. So I'll go ahead and close this question. I thought I would share with you a little bit about why I love chemistry and the applications and the research that goes on at the University of Minnesota. All right. And so I think the most important application of organic chemistry is in pharmaceutical sciences and the development of drug molecules. All right. Everyone is motivated by something different, but that's something that really is important to me. So I'm going to introduce you to a new functional group today called an aryne. So we've seen aryl groups, and these are kind of different than that. So a little bit about how I got here. I'm just a normal student. I was a normal student just like you all. So I was an undergraduate. I graduated with my degree, an undergraduate degree in 2011 from a school in California. And I went to the University of North Carolina at Chapel Hill, and I did my PhD there. It took 5 years. Then I went to the other U of M, the University of Michigan. And I started here in 2019 as a professor. So I have a research group here. And these people are the ones that are my PhD students and my postdocs and my undergrads. You may notice a familiar face right here. Lars, your beloved TA Lars, all right, is the one that he was a research student, an undergraduate researcher. He and I published a paper together. Aryne Plashek, who you haven't met, she's the one that developed all of your OLS packets a few years ago. All right, so these students are graduate students and undergraduates that are all here at the U. We're funded by the National Institutes of Health and the National Science Foundation, as well as some pharmaceutical companies. And so, yeah, these things are, science funding is incredibly important. All right, so the big picture for me, this is really what got me into science here, was the fact that all of these organic molecules, so agrochemicals, we live in the Midwest, a lot of things are, food is produced here and you need agrochemicals. It's estimated that actually only 3 billion people would be alive today if we didn't have synthetic molecules and synthetic chemistry. One of those look to your right, look to your left, like half the people would not be here if we didn't have synthetic chemistry and chemistry to make agrochemicals. This is a blood thinner, an organic molecule called Eliquis. This is a little bit more complicated. Both of my parents take this molecule, this pharmaceutical, and it keeps them alive. It prevents blood clots. And then organic solar cells are also a really big application of organic chemistry. So if you're interested in sustainability, it's very important to sort of understand organic molecules. So I'm going to talk to you today about this particular functional group, all right? So we have seen arynes, but we've never seen this functional group, which is called an aryne, all right? Turns out you can actually put a triple bond like an alkyne into a benzene ring, all right? And you can imagine that this is a highly reactive molecule. So this is very electrophilic as a molecule, all right? So this is going to react with a lot of different types of nucleophiles, all right? We don't need to worry about a leaving group here. We have this very reactive triple bond. You talked in 2301 about ring strain, and you talked about hybridization, right? An alkyne is usually an sp3 hybridized carbon that we see here in the alkynes. And now we've bent it into a benzene ring, all right? So it's very, very reactive for that reason. All right, so we can do many types of chemistry with a benzyne. So we can take this nitrogen and it can attack our benzyne bond. And the really cool thing about this is we have all learned about electrophilic aromatic substitution all the time. We know that a methoxy group, an electrophilic aromatic substitution, is going to direct to the ortho position, all right? So a benzyne allows you to get around that and actually directs to the meta position on a benzene ring, all right? So it switches the selectivity that we're able to get through electrophilic aromatic substitution, all right? Okay, that's pretty cool. Well, we can react. So we haven't talked too much about multi-component reactions, but we have two different termini of this triple bond. So what happens here, I should say, is we attack that with a nucleophile, and then this triple bond gets broken to put a negative charge at that carbon, and we can harness that and do a multi-component reaction. All right, so that's pretty interesting. We can use one molecule and do two transformations on it. Right? You learn things about like adding across alkenes, like hydroboration. We're only adding a hydrogen, right? So here we can have many different types of functional groups, right? Rarely in this class do we ever react and break a carbon-carbon bond, but you all know that the tautomer of this molecule is an enol, and that soft nucleophile of our enol is actually able to go ahead and add to our benzyne bond, all right? So these are really, really, really reactive intermediates that we don't get a chance to talk about in class, but you can break things like carbon-carbon bonds, right? Using intermediates that you've seen before, but these are gonna react in a different way, all right? Here's a super complicated molecule. What if this was on the final? Don't worry, it won't be. But right, how do we make that really complicated molecule, all right? Well, we can add, do a 4+2 cycle addition. You all learned about the Diels-Alder reaction last time, or in 2301 last semester, and we can go ahead and do a 4+2 cycle addition with this intermediate. So this intermediate can do so many different types of things, all right? So you say, well, oh my gosh, that's so great. Why didn't we learn about this really reactive, really useful functional group? Like, why didn't we learn about this? Why is this not, there's like one blurb, there's a paragraph in your textbook about this molecule. So why didn't we learn about it? Well, you all learned that sometimes chemistry can go off the rails and it can do reactions that you don't want it to do. All right, and there's a lot of drawbacks as to why we don't use a benzyne. All right, and this is where research comes in. So I'm going to show you the synthesis of the benzyne intermediate and the natural product synthesis of this molecule, elliptosine. This is going to tell you everything that's wrong with benzyne chemistry. And then I'm going to tell you how we solve those problems. Because that's what research is. You have to identify a problem. Look, we can make all these things that look like drug molecules, but it's really hard to do, okay? So here's the first problem, is that I told you this was really, really reactive. Right, this benzyne, here it is, and a pyridine was super reactive, but so we can't weigh that out on the bench top. We can't add it to our flask in our reaction. We have to make it and generate it in situ, all right? It's too reactive, so we have to actually make it in situ. And how we react it is with this how we make that is with this highly explosive molecule. All right. That's not a very good idea. We don't want undergraduates, graduate students, anybody to use really explosive reagents. All right. An explosive reagent usually has a really high ratio of carbons to nitrogen. So this is probably why you've never seen a benzyne, because it could explode when you're trying to make it. The other problem is the reagent that activates this to make this is lead, right? You see in the news all the time, lead pipes. Those are really bad for our water system, right? So we don't really want to be adding lead to chemical reactions, especially if this pharmaceutical is going to go into a patient, right? So we have to overcome that challenge of having lead. All right. What's another challenge? Well, I told you some really nice things about EAS selectivity. We can react in the meta position, but if this is an unsymmetrical molecule, you end up with two isomers, right? Just like in EAS, we have the same selectivity problem where if we had a methoxy group, you could add at both the ortho and the para positions, all right? Then you would have to separate those out. That would be challenging. Well, benzynes are also problematic because they're able to react if they're unsymmetrical and form multiple isomers. So reason number 2, we don't see these in our textbook. So another way that we can make this molecule is instead of having this selectivity problem, we could have an electronically biased molecule here, right? We could take something like this methoxy and maybe we could block these groups with a tert-butyl group or something, then it would only react at the para position, right? We kind of know how to get something selective. So we could use this molecule, this benzyne intermediate here, instead to be able to actually generate what we want, all right? We can kind of block the other positions and have this electronically biased, but it turns out that's impossible to make, all right? I talked to you about ring strain in a 5-membered ring with the triple bond. That's a no-go, all right? Actually, 120 years people have been trying to make this very molecule and completely failed at it because there's too much ring strain built up. So you say to yourself, well, this seems like a really terrible intermediate. Why would you show this to us? Well, that's what research is. Research finds problems with explosive reagents and molecules you can never make, and then we figure out how to do the impossible, right? We figure out the challenge, and then we form a hypothesis, and then we test our hypothesis to overcome some of these challenges, all right? So I'm gonna walk you through. These are the students on the team. Sal here has actually served as an OLSTA in past iterations of this class. Maybe you even had him if you've taken this a couple of times. Chris was the head TA at one point for your organic 2311 lab, so maybe you know him. And Felicia could maybe be one of your TAs now as she's a 2311 TA. All right, so these are the graduate students that do this research in my research group. Okay, so before I told you that we can have explosive reagents, and people have made slightly less explosive reagents, but they sort of don't overcome the challenges that we need them to, all right? So all of these ways to make benzynes that people have done research on just sort of don't fit the bill for a nice reagent, all right? We see a lot of, this is another explosive functional group, this is explosive functional group. These are really difficult to make, so this is not a solved challenge, all right? So what my research group did is we designed an ideal reagent to make a benzyne. We said we want to be able to make this in one step. We want to change this R group in one step. All right, those are our goals. This over here takes about 6 to 8 organic steps to make. So don't worry, that would show up on the final for the multi-step synthesis because it's super, super hard to do, all right? Think about 8 steps to make that one molecule, all right? So how do we know this is not explosive? Well, we can actually test reagents for their properties of explosion, all right? So we did this assessment. We looked in the literature and we said, how do we make sure this isn't explosive? Well, we did something called an Oreos plus analysis, and we took a look at some measurements of this molecule to make sure that it wasn't explosive. All right, so it's very important when all of these other molecules to make a benzyne are explosive. It was very important that we designed in sort of more of an engineering thing to design in that what we're making is not explosive. And so we did this analysis and it's not. All right, great. So I talked to you about how difficult the other benzyne precursor is. I'll show you the synthesis of this really quick. All right, so we have to use tert butyl lithium. We have to use strong electrophiles. We have to use harsh bases, multiple steps. You all have seen these arrows, right? Do you really want to do a multi-step synthesis of this? The answer is no. It's very hard to do, all right? So this is kind of one of the things that we were overcoming. We said we want to take all of this and we want to do it in one step. It's a really hard thing to do, but that's what research does, okay? So that's what we did. So we went ahead and we said that we can make this benzyne intermediate from this, which we can buy from a chemical company. We can use MCPBA. You all learned MCPBA is for epoxidations, but all MCPBA is an oxidant, and that's what we're doing. We're oxidizing this reaction. We go from a homogeneous solution to a heterogeneous solution. We add in, and this is a EAS reaction, right? We learned about electrophilic aromatic substitution. Turns out that this is a really nucleophilic carbon here due to these electron donating groups, right? And so we can do an EAS reaction on this iodine here, and we can go ahead and make our reagent in one step, all right? So we've overcome the challenge of having many steps to make this reagent. We can do it in one pot, in one reaction. So that was one. It's not explosive. We can now make this in one step. And then you see this particular fluorine here. What did we learn about fluorine? It's a great leaving group for a nucleophilic aromatic substitution reaction. All right. So we had this hypothesis that we could undergo, SNAR is another way to say NAS. We could undergo an NAS reaction and do an NAS reaction to form a bunch of nitrogen-containing aryne precursors, all right? So we looked at the top drugs that are FDA approved, and we chose all of these nitrogen heterocycles to add in for our reaction, and now we can add actual drug molecules. Some of these are allergy medicines, some of them are antidepressants. We can actually take these drug molecules and we can use them in our NAS reaction, right? And then we can start to make all of these different benzynes, all right? So that was our first research problem we needed to overcome. In order to actually do research on benzyne chemistry, we needed a good reagent for it that wasn't explosive, that was super easy to make with reactions you all do in this class. Everything to make this reagent is what we did in this class, all right? So my goal and hope is this is what students learn in 2302 in a few years, is this now becomes a textbook reaction, all right? So we learned about pyridine as a base, but pyridine is the number one aromatic and heterocycle in FDA-approved drugs. These are all drug molecules that you may actually take or someone in your family takes. And so is the question, can we incorporate pyridine into these benzyne precursors or aryne precursors? And the answer is yes. Okay, so this is research at the U, right? We identify a problem in organic chemistry, a reagent that's really bad and that can't be used to make drug molecules. And then we form a hypothesis, we identify what the problems are, and then we're able to overcome them using research, all right? The second mini story that I'm going to tell you about research has to do, again, with making drug molecules, all right? Look how complicated all of these molecules are, right? Many of them are made with 2302 reactions, but also we have a lot more complicated molecules, all right? So we thought to ourselves, what if we can make these complicated molecules through a benzyne intermediate? We do retrosynthesis all the time, right? So to make Ambien, which is a sedative to help people sleep, we could think about the retrosynthetic disconnection and have an aryne, that strange triple bond, react it with something like an organometallic reagent, some sort of carbon nucleophile, so maybe a phenyllithium or a phenylmagnesium bromide. Then we could take some electrophile, maybe an alkyl bromide, and do an SN2 reaction on that to make all of these drug molecules. But I told you in the beginning, we can't make any of these molecules, all right, because they were inaccessible due to that ring strain, right? From that 2302 or 2301 chemistry you learned, we can't have a lot of strain in rings, all right? We also learned this about lactam formation and lactone formation. We can't have these tiny rings because there's too much strain. So my group hypothesized that if you add a metal, I told you I'm an organometallic chemist, this entire D block. So we add a metal and we bind it to our triple bond, and that alleviates the ring strain, all right? So for 120 years, people tried to make these molecules, and no one thought about doing research on adding metals to alleviate ring strain, all right? So this is my research group and the faces of the students that work on this problem. Maybe you've met Ali before or Douglas. They're currently TAs in 2311, so maybe they're your TA, I don't know. But they help contribute to this particular research, all right? So we can deprotonate. You all have seen LDA before as a strong base. All we need to do is deprotonate and quench with a boron. We've learned a little bit about boron in our borohydride reagents, and we can use it for other things as well. We can now use metals to put these into our triple bond so that we can alleviate this ring strain. So we can use nickel, which is element number 28. We can use some phosphines. We've seen phosphine before, triphenylphosphine in our Wittig reaction, so be sure to study that. And we can start to see, we can get crystal structures. This is basically a molecular picture. We can take a compound, bombard it with X-rays, and those X-rays will bounce back where there are atoms, and that gives you a picture of a molecule, all right? So this is a picture of what this molecule looks like. We also use NMR, which is a big topic in 2301. NMR we can use are actually phosphine NMRs. So I know everyone thinks it's useless to learn NMR in 2301, and it's really complicated in 2311. But actually we use it all the time in the research group to make sure we know what we're making. All right, so we can go ahead and make for the very first time these inaccessible arynes. So it was back in 1902 that someone hypothesized this could exist. And this was published by our research group in 2024, all right? Over 120 years, people have been trying to make these molecules because they would be so good for drugs. And so we were able to do it here at the U for the very first time, all right? So this is the type of research. This is why I'm slow to reply to your emails, because we do a lot of research here at the U, a lot of really, really interesting things, all right? So how does this work in drug development? Who knows how to make a drug? It's actually pretty hard. You have to make maybe 20,000 molecules, all right? So you design a drug, you make it, you test it in some cell lines, and then you analyze your result. This is how those pharmaceuticals, even something as simple as Tylenol, right? This is how that was made, all right? You design it, you make it, you test it, you analyze it. So how do we know which drug to make? Well, I told you they may make 20,000 molecules, and so they'll find something that looks pretty good, all right? So this looks pretty good. It's able to, it's a kinase inhibitor, so it's able to have a lot of biological function. But what they found is when they added one nitrogen, they added a pyridine ring, that this worked 300 times better. One nitrogen changed that, all right? So this goes from taking maybe 2 grams of those giant horse pills down to like a tiny, tiny pill if you can just add one nitrogen to your molecule, right? This is how drug development works, okay? But how did you find out the nitrogen needed to be at this position? Well, they needed to walk it all the way around the ring, all right? Turns out if they put it at this position, gets worse, all right? Gets a little better, a little better. This is when it has a huge effect. Just putting the nitrogen at that one position, all right? So this is how drugs are made. They tweak teeny tiny places on it. This is why you need so many reactions in organic chemistry. So here's how they made those, okay? They did all of these different coupling reactions. So in order to make an indole scaffold here, they had to make an alkyne and then a coupling partner. Then to put the nitrogen at the 7 position, they had to do a reaction with an aldehyde. To put 2 nitrogens here to test, they had to use a nitrile functional group. It's really hard to make molecules. You might think these look really similar to each other. You're just changing the number of nitrogens, but we need so many types of reactions to do that, all right? So my research group said is, what if you don't need so many types of reactions? What if you don't need to make alkynes and ketones and nitriles and heterocycles? What if you can just have your coupling partners? Maybe you wanted an alkyl bromide as a coupling partner. Then if you had this new functional group, a new functional group for over 120 years, we wanted to use this functional group, never could. It could significantly speed up drug discovery. All right. And so that's what my research group has been able to do, is we've been able to take this common set of coupling partners and a really reactive alkyne and be able to make all of these types of drugs. All right. So here's an example with Ambien, all right, where we can swap our scaffold of Ambien to make sure it's the correct structure that we want, that's the best for patients, doesn't have long-term side effects that are really bad. We can just swap that out using this new chemistry invented just last year at the University of Minnesota. All right. So this is what we do here at the U. I think a lot of times people only think of it as a place where you get taught, and it's a great place to learn and for teaching, but it also does a lot for things like people developing drugs and needing added medicines. All right. So I just wanted to share with you, why I care about chemistry, why it's important to your lives. Maybe you don't take any of these molecules today, but most people will end up taking a pharmaceutical at some point in their lives, right? Maybe it's a treatment for cancer, or a treatment for diabetes. All of these are made by people that took this class when they were undergraduates, and they were inspired by it, and they decided to continue to pursue that field. So, all right, that is all that I have for you today. I just wanted to share, this is the last time I'll be able to teach you anything. So hopefully you can see how chemistry can actually be more exciting than just multi-step synthesis that, doesn't seem to be directed at anything, but instead it really is inspiration for making life-saving molecules. All right. Well, I really enjoyed this semester. Best of luck studying for the final for you all. I'm rooting for you to do really well. So it's been, this will be my last time teaching this class for a while. So I really enjoyed sharing what I know with you all.